Well, good morning or good afternoon, depending on where you're situated. This is the second day of the Opportunities and Tools to Investigate Occupational Factors for COPD webinar. And it is sponsored by the Respiratory Health Cross-Sector Council of the National Occupational Research Agenda. My name is Paul Hanneberger, and I'm glad you're with us today. We're in for a very good session. I'm the Senior Science Advisor in the Respiratory Health Division and co-chair of the NORA Respiratory Health Cross-Sector Council, and a member of the organizing committee for this webinar. And the other organizers, there's three other organizers, including Tom Croxton with the National Heart, Lung, and Blood Institute, Rafael de la Hoz, who is at the Icahn School of Medicine at Mount Sinai in New York City, and he will be moderating this session. And the other committee member is Lisa Posto with the National Heart, Lung, and Blood Institute, and she'll be moderating the section that starts later at 1:30 Eastern US time. I think we're ready to move on to the first presentation, and I'll let Rafael let you pick up from here and move on with the session.
All right, so I'll be brief. Welcome everyone. Thank you for attending. Please spread the word to your colleagues about the availability of this webinar on the internet for I don't know exactly the amount of time, but certainly several weeks or months. And I am Rafael de la Hoz. I'm occupational pulmonologist at the Icahn School of Medicine in New York City at Mount Sinai. And I'm actually also a member of the cross-sector council that sponsored this, organized this webinar. And as an anecdote, this was born of an initiative that I was trying to sell to, or to include in the programs of the the two major respiratory societies, the European, the American Thoracic Societies, unsuccessfully. So actually one of the criticisms was that the speakers, many of whom are speaking today, actually were too well established. And I said, well, if that is a fault, let me enjoy it in a different venue. And this morning we are concentrating on basic mechanistic aspects of this topic of work-related COPD. Dr. Christopher Carlston, who is a professor and chair of occupational and environmental lung disease at the University of British Columbia in Vancouver, Canada. And Dr. Farah Heratman, she is a professor of medicine in the pulmonary division at the Baylor College of Medicine. And as I said, please send your questions, as Dr. Henneberger said, and I'll try to definitely make sure that all of them are answered. And so we'll turn it over to Dr. Carlston. He's talking to us from the University of British Columbia. Chris.
Thank you. And I hope I don't have any connection problems. I've had none until suddenly I heard Dr. Barnes slow for me. So if I'm the only one that had a little bit of slowing of him, I'm slightly worried, but I think it'll be fine. I mean, I'm on a hardwired major university platform. So anyway, thank you very much, Rafael, Paul, etc. I found yesterday fantastic, and today is no different, and it's always a pleasure to hear from Dr. Barnes. And in fact, I think the connection from his talk to mine will be excellent on a number of levels, including the concept of accelerated aging, which I don't speak to specifically, but I think I can start by a tentative conclusion that the concept of combined exposures that I will speak to essentially further accelerates that aging. But you'll see that my presentation is much more speculative and perhaps conceptual, because the idea of combined exposures, as I'll show, has just been less studied, in large part because it's more difficult. But I think because of that, I won't have the level of detail that Dr. Barnes so finely outlined. Anyway, there'll be some great crosstalk, I believe. Oh, I didn't mean to skip that so quickly. I have no relevant conflicts of interest or financial disclosures to declare for this talk. So a high-level concept that I want to share, and perhaps it can be part of a discussion later, is that policy, and by this I mean policy regarding occupational or even in the more public health domain, exposures on various levels demands plausibility. And by that I mean essentially what Lindsay Stanek said best over 10 years ago, that toxicology, epidemiology, and controlled human exposures, I bolded, but she stated as part of this triad of data that together provide this foundation, and again, I'm underlying, of science that are used for regulatory decisions. And those in the US know that this has been exceedingly the case when it comes to air pollution regulation, in particular, being challenged by industry and others. They turn to the science, and we in turn turn to the science to defend decisions based on plausibility. That being said, I'll speak to more traditional mechanisms in the same overall vein that Dr. Barnes did. But I think there are other mechanisms at play here that are less traditional, including the behavioral and industrial design, cultural factors that are influencing all of what I'll speak to, but is yet another level of complexity beyond this. So what I just said is essentially now just graphically depicted for those that like to see it this way. And just to reiterate that we can divide the data roughly into these categories: the observational data of epidemiology, which can get huge numbers of people and lots of data, but is subject to the vexing issue of confounding and residual confounding. Animal and cellular research, which of course can lead to very detailed dissection of pathways and all kinds of neat methods to block pathways, etc. And then controlled human exposures, which are a smaller sample size and very limited in its own way, but has its own niche that I'll talk to. That's just the reason that we do this, and the reason why I'll show you we do this with combined exposures. I was most interested, this is an example of the epidemiology in this, when I earlier in my career was more focused on epidemiology, and noted that these children we followed from birth, and in this case just till age 8, although they were followed subsequently, when exposed to both typical allergens and typical pollutants, which you see listed on the bottom, in combination at high levels, had a markedly higher risk of developing asthma in their early life than either exposure alone, which conferred relatively modest risks, as you see in the blue and the red. So that's simply an epidemiological observation, not from the occupational setting, but that motivated other work from many investigators. And here's a summary, relatively recent, of some of the combinations of exposures, generally all highly relevant to the occupational environment, that together drive respiratory disease, generally focused on airflow. So a lot of work on FEV1, which I think you're familiar with by now. And also adaptive immune responses. The Diaz Sanchez in Southern California did great work showing how specific IgE would be driven significantly higher when in the context of diesel particulate matter, etc. And you can see, of course, going back in time even further, the classic asbestos and cigarette smoke combination, which markedly increases lung cancer risk. But the point I really want to make about this is that all of this had very little and continues to have very little attention to COPD specifically. So it's mostly been asthma, and as I said, lung cancer. But COPD, with the massive burden of disease internationally, of course, the fact that we understand relatively little about how combined exposures drive COPD, I think, is a real problem. And it's also, frankly, a real problem for my talk, because as I said earlier, had to speculate. But we will, I think, come to understand together how there are these, of course, diversity of exposures. And when you think about it, trying to study all of them in combination, you can quickly do the math, leads to quite a huge number. So we have to base our understanding, at least in large part, on basic principles, and then try to study as much as we can of specific common exposures. And of course, you're familiar with practically all of these, whether it's cleaners or pesticide application, bakers, miners, animal handlers, construction, etc. So what are the mechanisms? And again, presumptive in large part. Most of these have not been studied directly, in combination, and I think are not mutually exclusive. And many of these you'll remember from what you heard from Dr. Barnes, and I'm sure these are not exhaustive. So first, oxidative stress, I was gratified to see that, not surprisingly, really well established as perhaps the dominant mechanism of these exposures leading to COPD, as you heard. We didn't hear much about this previously, as I recall anyway, in the last couple of days, but the idea of susceptibility by genotypes, I think, is an important part of this, particularly because variants that are related to metabolism of oxidative stress are amongst those that have been the most well established and validated of all the genetic variants that are related to airways disease, including COPD. Something you might have heard less about, and I'll touch on briefly later, is host defense, and how host defense, whether it's antimicrobial peptides or the epithelium itself, which of course can be damaged by oxidative stress and other mechanisms, allowing access to the subepithelial layer and then downstream damage therein, that can be heightened in the context of combined exposures naturally. As I mentioned before, synergy with allergens, and so that was originally shown in the nose with David Diaz-Sanchez, I previously mentioned, and we've done a lot of work of that on that in the lungs, and primarily with asthma. But as I'll argue further later, the line between asthma and COPD is a fine one indeed, and I think some of these synergies are driving the concept of ACOS, or asthma that develops into COPD, and we'll get into that. Some of you may be familiar, others not. And finally, epigenetic effects. You heard briefly about HDAC with Dr. Barnes, but there are many others, and I think they're, again, this is highly on the speculative side in terms of them being fundamental to how combined exposures drive COPD, but I think there's a case to be made for this. So there is this diversity of mechanisms, and it's perhaps obvious, but nonetheless reasonable to note that this matches with the spectrum of phenotypes. So there's obviously a lot of attention now to phenotypes and endotypes, and trying to characterize an individual by their particular profile, cellular and otherwise. And it's likely that these mechanisms that we've been talking about connect to these different phenotypes, and then likely will help with treatment. Although, of course, we're by far more interested fundamentally in prevention. So on the right, you see the fairly well-established mechanisms from cigarette smoke, and I don't think any of this will be in conflict with what you heard from Dr. Barnes. Again, interestingly, oxidative stress or reactive oxidative species in the middle is central, and as you saw in much better and elegant detail from him, the various ways leading to that oxidative stress. And you heard already from Dr. Barnes how this can lead to MAP kinase and NF-kappa beta pathways that will lead, again, we're right in sync, he and I, with macrophages being critical, but he mentioned the other cell types, and I'm highlighting neutrophils for reasons that will become apparent, proteases, which you heard about. So this is all a brief recapitulation of what he said. But if you look on the left, you see occupational COPD, the established agents, or the better established agents, because there's certainly more, cadmium, coal dust, silica, etc. And you can see in parentheses the cell types, which are not exclusive, I think, but perhaps predominant in these, where they match clearly the same types of cells and phenomena that are listed with cigarette smoke. So this is a case where there are similar pathways, I believe, and I'm speaking of occupational exposures aside from cigarette smoke. Of course, cigarette smoke arguably itself is an occupational exposure, but if we consider these as occupational exposures and cigarette smoke as two different exposures coming together, these are some of the ways that they have common pathways, which likely would be an additive effect. And you heard from Dr. Blanc yesterday about, from the epidemiologic perspective, again, the crosstalk between epidemiology and experimental studies being critical. You saw that the data from observation is that many of these phenomena are additive. In fact, perhaps most, but there are some that are super additive for interesting reasons that we'll touch on and would certainly bear much more detailed investigation. One of the, again, factors that is compounding this and likely leading to super additive effects is the effect of genetic variants, which, as I said before, simply speaking to the glutathione transferases, which are probably the best established in the oxidative stress pathway in terms of their true clinical effect. Often because the glutathionase transferases are so commonly variant, the M1 in almost 50% of the population, so arguably not a variant, but equal to the wild type. Their effect is common to air pollutants, some of which has been established in the environmental literature, but these pollutants, of course, are occupational as well, diesel exhaust being a prime example. And this sketch from a review a few years ago shows some of the other less well-known, aside from the glutathionase transferases, and some less well-known in general, but to the scientific community that studies this, fairly well-established variants in MUC5B, for example, CFTR. You can look at the list. The TLR variants is something that Dr. Barnes has worked on, and we continue to, NF-kappa B, etc. But just speaking to one of these to show it in a bit more granular detail, we've shown that with diesel exhaust, CFTR activity is decreased, as you see in the red line here. And one can imagine that with variants in CFTR, that discrepancy, that loss of function and the downstream physiologic effects would be that much greater. Finally, as far as this slide, alpha-1 antitrypsin deficiency is genetically based, and the effect of alpha-1 antitrypsin deficiency was highlighted by Dr. Blanc yesterday. But this would increase the risk of COPD by losing inhibition of neutrophil elastase, so that neutrophil elastase, which you heard from Dr. Barnes, being more active would be one of the proteases that we think significantly increases COPD. I'm going to turn now to antimicrobial peptides, which I said earlier is probably something somewhat less familiar to this audience. And you see in this graphic, it's a little bit blurry, I'm sorry, that these are a host of peptides, really thousands have been identified, a handful of which have been more strongly correlated with clinical disease in earnest, and many more speculative in that regard. Whether they be antibacterial, antiviral, etc., have been shown in the smoking literature to be reduced. And one of the antimicrobial peptides that's best established as clinically relevant is LL37, otherwise known as cathelicidin. And that's been shown both in, again, smoking, as I just mentioned, but also decreased in occupational organic dust exposure. So there again is a common, perhaps not super additive, but a common pathway that would increase this phenomenon of loss of host defense, specifically in terms of antibacterial function, which, as you can see from this review, somewhat dated, but I think still generally correct and visually easy to follow, that this loss of antimicrobial function would lead, therefore, to more infections, which are obviously well established as critical to the COPD, especially COPD exacerbation, if not its original development. An example, just to give, again, some granularity to where an occupational exposure can indeed do this loss of antimicrobial function, is this data we produced, where on the far right, DE standing for diesel exhaust, that conferred a loss of another one of the well-established host defense peptides, alpha defensin 1, universally in these human subjects in this controlled study. That paper shows a similar effect in S100A7, which again is one of the better established antimicrobial peptides correlating with susceptibility for infection. So we can see, again, in a plausibility sense, that there are these background data that back up the concept that occupational exposures like smoking, and presumably worse together, increase the risk for infection, which is critical to COPD pathogenesis and progression, holding together this idea that infections are indeed an important part of the story in this context. But I said before that the idea of ACOS may be highly relevant here, because most of the literature on combined exposures has been done in the context of asthma. And that, of course, there's still relatively little, but more in the context of asthma than COPD. So conceptually, with on the left, the asthma background that we've summarized in the oval, using, for example, again, allergens, which are, of course, very common to occupational settings, as I showed before, and diesel exhaust, which, of course, is very common. This whole progression from the early genetic factors, epigenetics included, through the cellular display that has obvious clinical effects, whether it's neutrophils, eosinophils, etc. All of that can be, at least conceptually, thought, if persistent, to drive this phenomenon of ACOS, where asthma develops into a more fixed disease, which we can call COPD. And the different phenotypes, as you see here in this graphic by Nirupama-Pucha, I added the work phenomenon, because you can imagine from what we know already that various workplace exposures can push any of these phenotypes towards the center of this more fixed disease along the pathophysiologic pathways that Dr. Barnes described. So I think that the workplace is a part of this ever increasingly appreciated ACOS phenomenon, which of course confers a much more difficult to treat disease, as Dr. Barnes said, due to steroid resistance, etc. Another way to look at that is this from Jim Hogg's group in New England Journal, that whether it's on the left COPD or on the right asthma, the normal physiologic loss, the inevitable loss that even fortunate healthy individuals have over the years, is of course accelerated in COPD and asthma with a different temporal pattern, but nonetheless, either way leading to ACOS, where just by loss of the normal reserve and increase in the fibrotic pathways we've been talking about, that becomes a fixed disease that again is refractory to much of our typical treatments, and of course that leads to a much greater burden of disease. The idea that this can be driven by pollution is relatively novel, but at least in this one example that we published a few years ago, there was a rather convincing signal for how air pollution would drive asthmatics into COPD. Again, one would imagine, of course, the COPD population, many of them are active or past smokers. And so there's a plausibility to the combination of smoking and pollutants, occupational or otherwise there. And there's a corresponding larger epidemiologic study from the Canadian Community Health Survey, where approximately 10% of those originally with asthma ultimately were defined as ACOS, so progressed into the more fixed disease. And the risk of that was rather impressively associated with increases in particulate matter. So I think that corresponds, or if not a direct validation, is consistent with what we showed in 2016. So again, it's not pure speculation that the workplace would be associated with this ACOS phenomenon. Finnish colleagues that many of you know showed in this paper not so long ago that electrical and electronic production workers had a rather astounding increased risk of ACOS, of course, with a somewhat large confidence interval, but still impressive, and likewise fur and leather workers, as you can see. Now, we've talked a lot about COPD in general, but of course, COPD progression leads to a higher burden of symptoms, hospitalizations, economic effect on the countries that fund the system. So it's important to try to understand this progression and, of course, slow it down, if not eliminate it. Much of that is believed to be mediated by neutrophils. Of course, it's much more complicated than this. It's also somewhat controversial, but the neutrophils are undoubtedly important. How absolutely central relative to other cells we can leave aside for the moment. But there's an inverse correlation with the percent of neutrophils and this crude measure, but still commonly used as a basic measure of lung function, FEV1. And you can see that inverse correlation is pretty strong. The details of that are increasingly thought to be significantly related to the concept of nets, which are neutrophil extracellular traps. And I'll show you a picture of that momentarily. But as you can see here, and other studies corroborate this, the degree of netosis is significantly correlated not only with COPD in general, but with exacerbated COPD, which of course is in fact the #1 predictor of COPD progression, being the number of exacerbations and severity of those exacerbations. So we showed in the context of diesel being a common occupational exposure in vitro on this slide, and I'll show you in vivo subsequently, in vitro, that neutrophils, when exposed to diesel particulate matter, would in fact produce these nets, which you see down here. And these nets are quite rich in proteases, which again adds to the plausibility of the proteases that you heard from Dr. Barnes, leading to COPD. And the quantification of this phenomenon is shown here with the diesel particulate matter, again stimulating netosis. So again, we like to show in a safe, controlled, and ethically monitored on multiple levels, controlled human exposures in our lab. And in this study, this was the first ever to include this range of phenotypes in a controlled pollution exposure. Markedly, we thought, including ex-smokers who didn't have COPD, so we could include and compare them to those ex-smokers with COPD. We use a typical double-blind crossover study, as you can see briefly schematically here. But importantly, in terms of nets, we showed that near universally, these individuals had a marked increase in nets in their lungs. So no longer simply in blood in vitro, but in living humans in their lungs, leading again a higher, arguably, level of plausibility to the importance of this in these occupational settings. Now that netosis, as I said, was shown in smoking, was shown with an occupational exposure such as diesel. But those neutrophils not only perform netosis, but they also are activated in other ways. And the surface markers you see on the left of each of these panels are canonical for neutrophil activation. But importantly here is that this is most pronounced, in fact, only in terms of statistical significance in those with COPD, which are the darker here. So what we're showing is that of these three different groups studied in vivo in our lab, that those with COPD are the ones and only those that have an increase in activation of neutrophils, corresponding to cigarette smoke phenomena that you see here under different circumstances, but still with activation of neutrophils on exposure to cigarette smoke. So this study that was published in the Blue Journal, again in former smokers, showed a couple things, both highlighting COPD as a particular risk for progression in the context of occupational exposure, specifically diesel exhaust. The list here of serum amyloid A, one of the metalloproteinases, perhaps not as historically central as MMP9, but nonetheless plausible. C-reactive protein, very well established inflammatory marker, and VCAM1 were only elevated in those with COPD, again, former smokers. So if you consider smoking not only active, but as Dr. Barnes said, former smoking clearly leading to an ongoing inflammatory phenotype even after that smoking has ended. The markers MMP7, another metalloproteinase, and the very important, as you now well know, alpha-1 antitrypsin, were elevated across the three groups, but more elevated, so elevated to a greater extent in those with COPD. And I'll probably skip the details about the airway microbiome, but it's just to say that there's increasing evidence that our endogenous airway microbiome, what we walk with and live with on a daily basis, will change the response to air pollution. So the idea that air pollution is actually changing our antimicrobial function may explain some of the response to air pollution, including the responses that are COPD relevant. So we're back to this and the idea that, again, much more high level than Dr. Barnes, because the data is modest and there's more need for speculation, perhaps. Inflammation, neutrophil activation, and an imbalance in protease, anti-protease ratios has been shown, particular to those in COPD. The data on the loss of antimicrobial peptides, unpublished in the context of COPD. I showed you some from asthmatics, but we haven't published this in COPD, that we have presented that data in the ERS just a month ago. This is my last slide, and it is either inspiring or concerning in the sense that I've shown you mainly two exposure paradigms. So sure, moving from one exposure, which is extremely limited, to two, which shows almost universally that the phenomena, whether it's epidemiologic or in cells, etc., is heightened when we look at two exposures versus one. Of course, we're really living in a mix that's much greater than two exposures. So how to understand that, how to model it, and in particular, hopefully something we could get to in discussion, how we can integrate that into a compensation system where I have, and I'm sure others, hard enough time explaining how COPD can be driven by both smoking and other exposures, so that we don't just simply dismiss the smokers as being a smoking only disease. Imagine when we try to talk to compensation systems when the number of exposures gets even greater. I think that's a practical challenge to face. We lay out some of the conceptual ways to address that challenge in this paper, but I think doing this within a compensation system that doesn't necessarily respect highly kind of complex arguments, understandably, is going to be a challenge. So I'll end there and look forward to the discussion.
Yeah, thank you very much, Dr. Carlton, for that excellent presentation. And we are going also to the next presentation by Dr. Farah Heratman from Baylor College of Medicine. Here she is. Welcome.
Hi. Are you guys looking, are you seeing my slides and hearing me?
Very well, yes.
Excellent, thank you. I want to thank the organizers for inviting me to speak in this distinguished panel. And so what I'd like to do is, what I was tasked to present is really insights from and future directions for animal and cellular models to study work-related COPD. And so with that, I want to first tell you that I don't have any disclosures related to this presentation. And I'm going to tell you my talk objectives. I'd like to start by telling you a story about particle-based exposure that recapitulates human emphysema. And again, this particle exposure is really nano-sized elemental carbon black, and I'll tell you how we got there. I want to also tell you some mechanistic studies that link particle exposure and cellular dysfunction. The reason for that is really perhaps to start connecting together the ideas that we heard yesterday from the epidemiologists studying this occupational lung disease with something that perhaps can be examined, or some hypotheses that could be tested in the laboratory setting. And finally, what I'd like to do is kind of discuss with you future directions in particle-based exposure to lung diseases. And I don't want to just say COPD, because as you will see throughout the talk, or hopefully what I can communicate with you, is that it's going to be beyond COPD, beyond emphysema, because there's going to be some significant changes in the lungs in response to this particulate matter exposure. So with that, I want to start telling you how I got here. And this is an honest truth way of me telling you what it really happened. That is, you know, in two of my talented graduate students, a senior graduate student, Dr. Ming Sheng now, and Ran, many years ago, it's almost 10 years ago, kept on asking me, what is the dark material inside these cells? And so what they were referring to is that whenever they isolated lung macrophages from human lung to study their immunological studies, they were made this critical observation that whenever they isolated macrophages or lung antigen presenting cells extracted using CD1A marker, which there are a lot of it in active smokers and in patients who've smoked a lot, they found this dark material residing within these cells, and there is shown a representative here in this histology. And of course, armed with my medical knowledge in medical school and with pathologists who would help me, I basically dismissed their question. I said, you know, you just move on and please just focus on the immune cells that you're extracting, because what you're referring to is most likely what pathologists call antiprotic particles that are clearly associated with human smokers, and that they are actually just, you know, you could find them in hilar lymph nodes, and there are benign markers of active smokers or extensive exposure to cigarette smoke. And you have to understand one thing, that Ming had 3 first author papers and was about to leave my lab, and somewhat emboldened by his amazing talent, and decided to just dismiss me. And in fact, the next thing I know is that I received a phone call from a professor of material science and nanoengineering at Rice University, who basically stopped me and said, Farah, what are these human samples that your student gave me? And using Raman spectroscopy, we find that what you have here is basically elemental carbon black. And so where'd you get that? And in fact, it's been unbeknownst to me that Ming and Ran had actually collaborated with our now well-established collaborators at Rice University to try to identify what these black particles were that were concentrated within the antigen presenting cells and macrophages. And it turned out that, in fact, now 10 years later, I can clearly verify through multiple experimental studies and published work that, in fact, human lung macrophages harbor elemental carbon black. and so shown here in this transmission electron micrograph. And so now I had to go back and try to understand what are we dealing with? What's elemental carbon black, how it's found in the environment. And so long and short research basically can find that in fact, pure elemental carbon black are nano-sized, and they are generated in response to incomplete combustion of organic material, which certainly found in cigarette smoke, but it can also be found other sources that we routinely get exposed to. And that include coal miner workers, obviously cigarette smokers, exposure to biomass fuel, and of course, more commonly that we're dealing with is wildfires, as well as diesel. And in fact, if you look around you and there's anything that's you see that's black, most likely contains elemental carbon black, and that's made in the industry. But for more of the aficionados of the toxicologists, we've got a lot of pushback that these material that you found in human lungs are in fact most likely black carbon, which is different than the flavors of that we see in elemental carbon, because it's what they refer to as soot. Whereas carbon black, which is generally made in industry and in engineered products, which is used as nanomaterial, is completely different. And in fact, that they are referring as black carbon is what you're environmentally exposed to, whereas carbon black in engineered material is occupational. But whenever you get pushed, in fact, you push back. And in fact, working with our collaborators at Rice University, we indeed showed to many of the skeptics that in fact, elemental carbon black isolated from human lungs shows similar particle size and characteristics. And in fact, when you look at these TEM micrographs that are found from diesel, soot, nanocarbon black, whereas industrial made carbon black in the middle, it's very similar in every category that you'd like to do, including sizes, that is in the nanometer size, as well as the grape-like formation that elemental carbon black makes. And again, the one in the C panel is found what we found in the human lung. So emboldened by that and very much vindicated, Rand continued to follow the study. And in fact, she showed using, again, Raman spectroscopy and transmission electron micrograph that these cells indeed harbor carbon black. And in order to then verify that this carbon black that we found in the lungs is not because these patients were inhaling the very clean air that we inhale in Houston, Texas, but in fact, it is somewhat related to the material that they inhaled. So for that, she then exposed mice to cigarette smoke and isolated the very same cells or equivalent cells that we found in human, that this time is macrophages or antigen presenting cells as CD11C or SMK cigarette smoke and macrophages. And she showed that in fact, using same Raman intensity and spectroscopy, that it is very much recapitulates what carbon black reference does. And so indicating that there is somewhat an urgency for us to try to understand what is the function of this nanomaterial inside the antigen presenting cells in the lungs. So with that, we wanted to ask whether can elemental carbon black be used to model emphysema? And so to do that, again, this time with a real collaboration with Rice University, Dr. Jim Torr, we were able to not only get a hold of carbon black, which is nanoparticular matter, but also an altered form of carbon black, which is pegylated form, which changes completely characteristics of the carbon material and makes it more soluble. So it's no longer this hydrophobic penetrating within the membrane of the double layer membranes of live cells. And so we then used not a random number, but a number that we kind of back calculated from what we had identified in the lungs of heavy human smokers. And we came up with a design to then expose mice gradually to the total amount over time, and then rest mice for a week and then examine what happened. And here's a real picture of these lungs, and you can see that the nanocarbon black results in not only a very large lung volume, similar to, again, this is done using a CT scan, but also that the pegylated form of that actually abrogates the pathological effects that you see in or emphysematous changes that you see in carbon black. And here is shown in histology, again, the middle panel being the pure form of carbon black that we found in human lungs. And if you altered it, you made it pegylated, that it no longer resulted in a toxic form. And so we then found the same type of mediators that we had originally identified in human lungs, that is expression of MMP9 and MMP12, and activation of the T helper type 17 cells, that the canonical cells that produce IL-17, which then recruits alveolar macrophage, alveolar neutrophils within the alveoli, and again showed that nanocarbon black activates similar inflammatory mediators found in human emphysema. And of course, if you take then mice that are lacking IL-17, and these mice were significantly protected from the pathological effects of emphysema in this time frame. And so with that, then Rand went on to then discover, again, another critical observation, that when you looked at the bronchoalveolar lavage fluid from mice exposed to carbon black and now rested for a week, that you actually still see quite a bit of havoc in the lung, and that you see, as depicted here, in cellular damage, which clearly is in sort of a signal for the lung to know that there's danger, and that activates the inflammasome pathway. And so that is basically lacking the pegylated form of carbon black exposure. And what she found, that in fact, there's a significant amount of LDH release, again, indicating there's cell toxicity. And then there's activation of both IL-6 and IL-1 beta in the lung homogeneous, as mice were exposed to carbonated or nanoparticulate matter of carbon black, but not in the pegylated form. So again, we wanted to then, this is the beauty of working in the animal model, then you can actually push forward and then see whether the inflammasome pathways are critical in some of the inflammation that we have found. And indeed, we were able to get a hold of the adapter molecule knockout. These are called Pickard knockout mice, where they no longer are capable of activating the inflammasome pathway, the NLRP3 pathway. And when we took these mice and again exposed them to nanoparticulate carbon, we found that these mice clearly were protected, as shown here, by these mean linear intensity reduction, meaning that they don't have the emphysematous changes that we see in the wild type mice. But then it was also, we're looking at the BAL cellularity, we found that there is actually reduction in both macrophages and, more importantly, in the lymphocytes and neutrophils that we know are critical in promoting or causing the lung destruction. So what I'd like to tell you that we have now been able to now model together a way of finding these particulate matter found in the lungs of in patients with emphysema, and again use it to show that these are not benign particles as we have been told, or it's still probably in the textbooks that shows that these are non-pathogenic. So what I want to do next is tell you some mechanistic studies, that more recent mechanistic studies that links particle exposure and cellular dysfunction. And so by now, you know what kind of students actually get recruited to my lab. And so with the next study, Chen-Ying Chang decided to just follow the studies that Ran had initiated in the lab. And this time she actually made some critical observations herself and found that in fact, carbon black within antigen presenting cells and macrophages, as well as antigen presenting cells, alters their metabolism. And that's shown here, again, cells that are extracted from the lungs of mice that were exposed to carbon black induce interleukin-4 induced 1 gene, as well as arginase 1. These are two enzymes that are critical for activation of phenylalanine and arginine. And in fact, when she co-cultured these antigen-presenting cells with carbon black or without carbon black, and then co-cultured them with their CD8 T cells, these are the CD8 T cells that are critical for induction of emphysema, she found that they actually alters their phenotype. And so her question joining my lab was, how does carbon black alter immune cell metabolism, and what are the consequences of that? So in order to first dig into that, she took advantage of some data that was in the lab that we had shown that there is a, when she looked at the full genetic spectrum of genes that are altered in antigen presenting cells that are harboring carbon black, she found a very strong signature for IL-7, for interleukin-10, and some of the other chemokine receptors and some other pathways. And so taking that, she wanted to then validate these genetic findings. And indeed, when she took bronchoalveolar lavage samples from mice that were exposed to carbon black, she found a significant upregulation of IL-10. This was not, this was specific because she actually did not find a quantitative increase in TGF-beta. So how does IL-10 in macrophages get secreted or exposed, and when she dug into the literature, she found that in fact many many pathways that are important for macrophages, and when they're combating various insults in the lungs, increase IL-10 activation. And so in order to then sort of get more of a global picture of what is happening in the lung, she then went on to look at an unbiased metabolomic pathway in the whole lung of mice that are exposed to carbon black. And what she found, lo and behold, is that there are many pathways that are important in sugar metabolism, that is pyruvate, glucose, fructose, and lactate, are highly increased in the mice, in the lungs of mice that are exposed to carbon black. And she again validated the increase in lactate in a separate cohort of mice that were exposed to carbon black. Then to give it a more of a translational work, she then isolated lung macrophages from human lungs and exposed them to similar amount of carbon black that we actually find in the lungs of humans, and indeed found that the human lung macrophages also increase lactate production in response to carbon black. And finally, to verify that what she has found is in fact could be echolocated to the location, and she used a very high throughput technique called DESI, which is the desorption electrospray ionization and mass spectroscopy, and showed that indeed the areas of the lung exposed to carbon black corresponds very nicely to the increase in the lactate production. Again, putting it all together, she's now identifying that exposure to these particulate matter increase and the lactate production or glycolysis within the lung macrophages. And so what she wanted to know is how does carbon black change metabolic profiles in macrophages? And so to verify that, and again, we posed the question, are you sure it's the macrophages? You know, at the 30,000 feet view, it may actually be other cells. And so in order to answer that question, she then resort back to transmission electron micrograph. This is a beautiful image. You could see in the top left corner, a beautiful type 2 cells with the surfactant lamellar bodies and alveolar type 1 cells in the bottom, all hugged by a beautiful monocyte or macrophage in the lung. And this is what you see in the lungs of mice that are exposed to carbon black. Again, that same lung macrophage, well, you don't see any carbon black exposed in the type 2 cells or type 1 cells. You can see that that alveolar macrophages have very effectively gobbled up any lung carbon black within the lung. So then focusing again, making a critical observation here, when she looked at the mitochondria within the cell, what she found that while there was a accumulation of carbon black within the cytoplasm, that if you focus higher power, that you actually do indeed see that carbon black is residing within the mitochondria. And then calculating the actual numbers, she found that between 10 to 15% of mitochondria within macrophages are now embedded with these laced with these carbon black. And so this unfortunately brought us to several nightmares from medical school time, because now I had to go back and put my ideas together. How do we go from resolving these carbon black lodging within the mitochondria to metabolic changes? And lucky for us, in fact, we didn't have to search very hard, because what we found was that indeed two of the critical critical enzymes that are the rate limiting enzymes in metabolism of glucose in mitochondria are GLUT1 and LDHA, both of which are significantly increased in the antigen presenting cells that are harboring carbon black. And so in order to then really go one layer deeper into this, what we're seeing is that there's increasing sugar metabolism within these cells. Well, how does that translate into the real life? Well, in real life, most of our energy from mitochondria, in fact, is made through oxidative phosphorylation and not gluconeogenesis. And so when she used seahorse assay to then examine what happens to the macrophages that are laced with carbon black, she again was very able to use seahorse assay to show that, in fact, glycolytic capacity in cells exposed to carbon black are increased, and that the conversely, using oxidative phosphorylation assays in seahorse assays, she showed that maximum respiration rate in mitochondria are significantly reduced in cells that are expressed that are now harboring carbon black. And so to put the story together, then she went back to her, and again, to try to understand the mechanism of how this happens, then we have to resort back to our genetic studies, where she very nicely found that, in fact, antigen-presenting cells that focusing on the glycolysis pathway, antigen-presenting cells they upregulate many of these genes that are critical in the glycolysis pathway, angiogenesis, and cell survival. And if you put all of that together, these are critical genes that are downstream of activation of HIP-1 alpha, and that's hypoxia-induced 1 alpha. And when she exposed carbon black to bone marrow-derived macrophages, when she found that indeed in a dose-dependent way, HIP-1 alpha is increased in response to carbon black. And that we know that the HIP-1 alpha is critical in this pathway, or in this glycolysis, activation of glycolysis in this, because when she took mice and removed HIP-1 alpha specifically in macrophages, she was able to show that, again, these cells are now no longer capable of responding to carbon black, as shown here in a dose-dependent way, and they're incapable of increasing their lactate. So I know I kind of went in a rapid fire here, but I would like to sort of tell you in a way what I've just told you. That is, what we've shown you here is that elemental carbon black is not very different than black carbon that is found to be present in soot or other material, and it is indeed found within the macrophages and alveolar cells in human lungs. That these nanosized elemental carbon black exposure in the lung causes emphysema. We've shown that the physical and chemical properties of carbon black are critical in their toxicity, and that both innate and adaptive immune pathways are activated in carbon black, very much like what we saw or what my lab has shown earlier, that it's driven by Th17 cells and then an induction of the macrophages, as well as induction of MMP12 and MMP9. And that hydrophobic forms of the carbon black aggregates inside cytoplasm and can penetrate mitochondria, altering their metabolic pathways. Now, this is really critical, and this gets into the what I just told you earlier in the introduction, that I'd like to go tell you that above and beyond induction of emphysema, or all of the other pathways that I've just told you about that are that are activated in response to carbon black, they're altering cell metabolism. So this is really big news, and that this alteration in cell metabolism is very much driven by hypoxia-induced factor 1 alpha. And so what I'd like to tell you is that these sort of this new revelations are perhaps using a new foundation for us to go deeper into how are these materials that you're exposed to that launch into your lungs and are picked up by these innocent alveolar macrophages can now turn the system around and perhaps induce something that you unexpected, or something that you're basically were unaware of. And so what I want to leave you with, there are some questions that, again, I was tasked to kind of come up with to how can we take these ideas that are epidemiological based, that human based, and now with animal models put together in cellular and in vivo studies to try to come up with ways that we can take advantage of this information and then apply it in translationally. So the first question that obviously come to mind is that, are these findings that we've found are related to all hydrophobic nanocytes particles? And the reason I emphasize hydrophobicity is that because I showed you in the previous data that if you change the hydrophobic component of the carbon black or nanocytes particle, that they no longer are toxic. What I didn't show you when we published in eLife is that in fact the size also matters. So the smaller the size, the more toxic these agents are. So if you take it, to 15 nanometer to 70 and 100 nanometer, they're not as toxic. And that may actually be because of what we just unraveled, that is actually changing the metabolism of the cells. And what is the long-term effect of altered mitochondria on health effect? And I want to tell you the health is because, again, it is no longer from what we've shown you is that it's changing, just causing emphysema. It is changing a very important cell's metabolic pathways in the lungs. And how is that going to alter their ability to fight tumor, for instance, tumor immunity, antiviral, antibacterial, and how is that going to change? More importantly than what we've shown in previous couple of years is that macrophages also have a secondary function, that is they recycle surfactant in the lung. And so how is that going to alter surfactant biology that we've shown that these is a critical function of these macrophages? And finally, what I'd like to leave you with is, can we change cellular responses to particles in vivo? Again, we know that the inflammasome pathways are critical in this. And so can we think about ways to, again, harness this information to help in translational ways? So with that, I want to leave you. Of course, it takes a village to do this work. I want to thank my sponsors, which is National Institute of Health and NIOSH, the VA Department of Veterans Affairs, and as well as CPRIT Foundation. And I want to acknowledge the work that was done by my talented graduate students, and two of which I just emphasized to you. So with that, I'll stop.
Thank you very much for that wonderful presentation. And I couldn't have anticipated that it melded so well and follows so closely the previous two in terms of a logical development, but also how many questions it kind of generate. So To the audience, I just wanted to make sure, please, we have a discussion section now, and that in terms of time, we have a few minutes, until maybe the next 10 to 15 minutes, because we exceeded a little bit the time. So please place your questions in the question and answer, using the question and answer function of the Zoom platform. So I just wanted to, Dr. Karaman, I want, what is the, for something that is ubiquitous, right? That, and that we considered, yeah, I was in medical school and I remember having been told that, we just filter stuff and that it doesn't necessarily mean disease, not necessarily that it was desirable, but that it was found in the lungs of very healthy individuals. So what what critical factors do you think are identifiable or thus far in the in the making of these particles from disease causing and non-disease causing, because it ultimately, that is the question, right? If you do an autopsy and motorcycle accident, somebody young and no lung disease whatsoever, they have they can have, if they live in the cities, I remember that from medical school, that if they live in cities, their lungs are going to be loaded with those, and then you take individuals and whom these could be causing disease. So you have mentioned some of those factors, but maybe we should start the discussion by honing in on that. What is a critical factor?
Right, so I think that perhaps I went a little too fast because that paper was published in eLife. We have found that the physical characteristics of these particular matters, literally. That if you have their hydrophobicity, that if you actually pegylate these molecules, that then makes them larger, and they actually can stay in suspension in fluid, they can be picked up and cleared. And in fact, if you let these mice live long enough, you actually see that they are slowly cleared from the lung. However, those particular matters that are not hydrophilic, and that remain hydrophobic, and they are in nano particular size, they remain in the lung, and they actually get picked up by macrophages. Then they disperse, and then there's a second set of macrophages comes in to pick them up. So how is it that we don't see the havoc that we see in the lung of urban dwellers compared to a heavy smoker? I think it's quantity, really. It's the amount. So the fact that we cannot remove these things sort of points to the fact that that we need to then think about pathways that these cells are using to activate the destructive pathway that we showed. And so maybe go back to the drawing board and try to understand what is it that we can disrupt in order to start the healing process. So it's one of these catch-22s, right? It goes in the lungs. We know we can't take it out, but we know that there are pathways that's activating. So let's start with that. I don't know if anyone else in this panel has any ideas, but be happy to hear that.
Can you connect while they think of other questions, which might be many? Can you think of a connection, with the? I mean, you showed the anaerobic metabolism, right? That rise in lactate, and but I was hoping for, out of ignorance, of course, that it would connect with the oxidative stress mechanisms that we have referred to so much in emphysema, and the other, particularly oxidative stress, and some of the inflammatory cell path that we see involved. Of course, this is a story as usual of mice and humans, and there are always, but to try to navigate towards a connection to those more traditional mechanisms of pathogenesis of emphysema. Another one, the protease antiprotease balance within the lung. So how do you think, so what I'm getting at is your research bridging towards those, or do you see that or not? How's that? What is the perspective there?
Right, no, absolutely. So the same mediators that Peter and Chris presented and we have published on are certainly in play. The same mediators I'm talking about, we personally, or we didn't really look at their NLRF3 or the oxidative pathways, but certainly looking at the immune response, the induction of neutrophils, induction of newly arrived macrophages, which are then emboldened to activate many cellular pathways that we saw in the previous talks, and that is certainly in place. I would say that what I'd like to do is that this is really showing a new paradigm that cigarette smoking, that what we see that's being released in the air with every puff, that it contains not 4,000, certainly contains 4,000 chemicals, a lot of which then we have heard about how they activate reactive oxygen species, but that particular matter, or what's seen that's invisible to our eyes, that right in front of us, is the incomplete combustion of carbon material that is inhaled. Then now it makes sense that when you have women, for instance, in developing countries who are exposed to these biomass fuels, that they're inhaling a lot of these things that are just accumulating in their lungs, and a lot of them show up with emphysema. And so, again, from human observational study to animal, to then identifying how this has happened. So what I'd like to tell you that the story that I presented, the second part of the mechanistic studies, actually is a lot deeper, and I encourage you to hopefully by next week see it in Science Advances. We actually shown that carbon black induces tumors in mice, and we in fact have two separate models of adenocarcinoma, and then we were able to show that some of this changes in metabolism. So the carbon black in and by itself doesn't cause cancer, at least not in our hands. If you give it to wild type, they don't get cancer, but it changes the metabolism in antigen presenting cells that then allows them to then ignore or cause a quiescent state, where actually then allows the tumors to flourish and metastasize. So that's why I wanted to emphasize that, I talked to Dr. Postno, should I talk about cancer? And I, at the end of the day, for a 25-minute talk, I decided, why don't I just tell you mechanistically what happens with carbon black, and then just sort of leave the rest of the imagination. But I'll happy to share with you that it is very important that this changes in metabolic state actually drives tumors.
Right.
Something that we have seen very much goes hand in hand. Smokers with emphysema at a much higher risk of developing lung cancer. Again, it sort of nicely validates some of the human findings.
So if you were going to go out on a limb, if I go back to my medical school, and that's in decades, that's we keep getting smaller here, from particles that we can see to those that we cannot see, and now we are down, last I read a journal, and I'm behind with my reading, of course, like most everybody that I know. We were down to the 2.5 particles in the air pollution, and it seems that do you see us having missed the real culprits, or what are we with that, with the particle size in terms of pathology? Do you think that a 2.5 microgram micrometer particle is not as critical, that we probably it's just a surrogate?
But remember, Raphael, that
Yep.
PM 2.5 is 2.5 and smaller.
And smaller, yeah.
So it includes everything. So in this question of whether we should be regulating ultrafine versus 2.5, et cetera, I mean, it's been debated considerably, and there's never been a convincing set of data to suggest that. And I'm talking again about regulation. It's a bit different if you want to understand what is the critical size for pathology, but from a regulatory perspective, there's never been convincing evidence that separately regulating a smaller particle would ultimately change outcomes. It may be that it would, and it may be that it hasn't been studied sufficiently, and some feel passionately that we should be regulating ultrafine particles. But yeah, most important point is that 2.5 includes the smaller particles. So like in our studies, we titrate the exposures to a PM 2.5 level, but the reality is that most of diesel particles are between 50 and 100 nanometers. So our PM 2.5 is actually really tiny particles. So that's just important for everyone to everyone in the audience to be aware of.
And the other thing, you refer to carbon black, but these particles also, what else is in them other than that carbon?
Well, I think for us, carbon black is just carbon black, right?
It's elemental form, and it's a lattice sheet that they form in these buckyballs that they're basically grape-like. They form, so they're
very pure.
They are in the amount of it. Their size is about in nanometer, but in reality, for instance, what Cabot makes, which is a industrial form of carbon black, that is in the order of 50 nanometer. So there are subparticles there, but what we found in the human lung was pure elemental carbon black.
I wanted to ask Farah about connecting the dots here. Maybe I missed this level of detail, but when you talk about finding elemental carbon in humans, I mean, that's after it's been inhaled and processed and gone through multiple metabolic or metabolic, I should say, pathways. That's not necessarily what they inhaled. I mean, I'm not sure that finding after, ex vivo and through a whole process of putting it through spectrometry is really the same as what they inhaled. So I wouldn't necessarily, I mean, I don't know enough to say this strongly, but I don't know that I'd be confident that they actually inhaled at the point of inhalation pure carbon.
Well, I would say that they're most likely not. But what I didn't tell you is that what we did was we isolated the cells that are harboring this dark material and put them through proteinase K to remove any proteinaceous material, any other material that are with that. Now, then that material is what's sent for Raman spectroscopy. Now in correlate to that, when we did the animal studies, that's the eLife paper, we actually exposed mice to cigarette smoke, and then we extracted the cells, again, antigen presenting cells, and then put it through Raman spectroscopy and found a signature consistent with our reference carbon black. So I can tell you that, obviously we're limited to technical ways of trying to really decipher what are we inhaling. But I can tell you that that's lodged, and it's the fact that it's very small particle that is hydrophobic in nature that it can penetrate through a double layer membrane and lodge and actually form aggregates inside cytoplasm. And some of which unfortunately then gets migrated to the mitochondria. And that's how we see the change shift in the, so how does that happen? And what are the timeline of that? That's absolutely, again, open areas that should probably be investigated. And this is something that my lab particularly didn't do it. We just looked at the end result.
I'm kind of curious, like I was talking about how important our science is to policy. Does anyone know, and maybe you'd be in the best position for, but does anyone know how the carbon black research has impacted policy on the production and sale and occupational kind of protections, et cetera, for this product that's massively important to industry and our commercial enterprise?
When our E-Life paper came out in 2015, again, we got bombarded by people. In fact, there's a commentary that's very funny. If you actually look up the paper and look at the commentary that we rebutted, and that we got a lot of pushback from the industry that what you found had nothing to do with carbon black, and we were able to point by point rebuttal that. And our point of the paper was that at least in the industry level, occupational level, which some of these people that are making carbon black are getting exposed to, changing the physical chemistry of these grapes of carbon black by making them less hydrophobic, by increasing their size, perhaps could mitigate some of these in occupational exposure. And we got a lot of pushback, and I don't know if it got anywhere beyond that.
Do you think that the mobilization, of these particles to the mitochondria is a critical step in their pathogenicity?
Well, at least it's a critical step in altering mitochondrial function, yes.
Okay.
Their metabolic change.
But did you see less for emphysema, for example, as an end result in those animals that did not have that much of that mitochondrial migration?
Well, so in the first paper, we didn't re-examine all of that, but the first paper where we actually pegylated and enlarged the carbon black particles, we did not see the same metabolic changes as we saw in the.
So at least suggest that it could be.
Suggests, yes.
Exactly. All right. So I hope you keep having these brilliant students under your direction.
They ignore me, that's the best part.
And thank you everybody for their attention, the participants for their excellence, the lecturers for their excellent presentations. And we are going to close the morning session, and we have a very interesting afternoon one. So hopefully we'll have most of you there. Thank you very much.